Today we are going to learn some aspects related to antigen, antibody and MHC molecules. So first let's learn about antigens. Antigens are any substance that the immune system can recognize and that can thus stimulate an immune response. If antigens are perceived as dangerous, for example if they can cause disease, they can stimulate an immune response in the body. Antigens may be contained within or on bacteria, virus, other microorganisms, parasites or cancer cells. Antigens may also exist on their own, for example, as food molecules or pollen. A normal immune response consists of the following: recognizing a potentially harmful foreign antigen, activating and mobilizing forces to defend against it, attacking it, then controlling and ending the attack. If the immune system malfunctions and mistakes self or non self, it may attack the body's own tissues, causing an autoimmune disorder such as rheumatoid arthritis, Hashimoto thyroiditis, or systemic lupus erythematosus, the lupus disease. Then, what is immunogenicity? Ability to induce a humoral or cell mediated immune response. Then, antigenicity, ability to combine specifically with the final products of the immune response, secreted antibodies or surface receptors on T cells. Once again, immunogenicity, ability to induce a humoral or cell mediated immune response. Then antigenicity, ability to combine specifically with the final products of the immune response, secreted antibodies or surface receptors on T cells. Immunogenicity means ability to induce a humoral or cell mediated immune response. Antigenicity means ability to combine specifically with the final products of the immune response, secreted antibodies or surface receptors on T cells. Then determinants of antigenicity. Some of the determinants of antigenicity or antigens are molecular size, chemical nature, foreignness, physical form, antigenic specificity, species specificity, organ specificity, isospecificity, autospecificity, genetic factors, age, degradability, dose of antigen, and route of administration and adjuvants. These are some of the determinants of antigenicity or antigens. So, what was an immunogenicity? Ability to induce a humoral or cell mediated immune response. Antigenicity, ability to combine specifically with the final products of the immune response, secreted antibodies or surface receptors on T cells. And first, we just mentioned some determinants of antigenicity. The next, requirements for immunogenicity. Foreignness. An immune system normally discriminates between self and non self such that only foreign molecules are immunogenic. Then, size. There is not absolute size above which a substance will be immunogenic. However, in general, the larger the molecule, the more immunogenic it is likely to be. Then chemical composition in general, the more complex the substance is chemically, the more immunogenic it will be. The antigenic determinants are created by the primary sequence of residues in the polymer and or by the secondary, tertiary or quaternary structure of the molecule. And physical form. In general, particulate antigens are more immunogenic than absolute ones and denatured antigens more immunogenic than the native form. So, particulate antigens are more immunogenic than soluble ones, and denatured antigens are more immunogenic than the native form. Then degradability. Antigens that are easily phagocytosed are generally more immunogenic. It is because of most antigens, that means T-dependent antigens, the development of an immune response requires that an antigen be phagocytosed, processed and presented to helper T cells by an antigen presenting cell, APC. Then contribution of the administration. Dose. The dose of administration of an immunogen. The dose of administration of an immunogen. So contribution of the administration dose. The dose of administration of an immunogen can influence its immunogenicity. There is a dose of antigen above or below which the immune response will not be optimal. Then route. Generally, the subcutaneous route is better than the intravenous compared with intragastric routes. The route of antigen administration can also alter the nature of the response. Then adjuvant. Substances that can embrace the immune response to an immunogen are called adjuvants. The use of adjuvants, however, is often hampered by undesirable side effects such as fever or inflammation. Then immunological adjuvants. To enhance the immune response to a given immunogen, various additives or vehicles are often used. An adjuvant is a substance that, when mixed with an immunogen, enhances the immune response against the immunogen. So, adjuvant is a substance that, when mixed with an immunogen, enhances the immune response against the immunogen. Then adjuvants are substances that, when mixed with an antigen and injected with it, enhance the immunogenicity of that antigen. Then adjuvants are often used to boost the immune response when an antigen has low immunogenicity or when only small amounts of an antigen are available. Then adjuvants may be added to a vaccine to modify the immune response. Then types of antigens. T-dependent antigens. T-independent antigens. First we are going to discuss. T-independent antigens are antigens which can directly stimulate the B cells to produce antibody without the requirement for T cell help. In general, polysaccharides are T-independent antigens. The response to these antigens differ from the response to other antigens. Then size we just mentioned. Then dose, route and adjuvants. We mentioned some adjuvants. Adjuvants may be added to vaccine to modify the immune response. Adjuvants are often used to boost the immune response when an antigen has low immunogenicity or when only small amounts of an antigen are available. Okay, then types of antigens. We mentioned T-independent antigens. T-independent antigens are antigens which can directly stimulate the B cells to produce antibody without the requirement for T cell help. In general, polysaccharides are T-independent antigens. The response to these antigens differ from the response to other antigens. Then T-dependent antigens. T-dependent antigens are those that do not directly stimulate the production of antibody without the help of T cells. Proteins are T-dependent antigens. Structurally, these antigens are categorized by few copies of many different antigenic determinants. Then mechanism of immune stimulation by adjuvants. Mechanism of immune stimulation by adjuvants. Extend the presence of antigen in the blood. Extend the presence of antigen in the blood helps the antigen presenting cells absorb antigen, activate macrophages and lymphocytes, support the production of cytokines. Then mechanism of immune stimulation by adjuvants. Extend the presence of antigen in the blood helps the antigen presenting cells absorb antigen, activate macrophages and lymphocytes, support the production of cytokines. Extend the presence of antigen in the blood helps the antigen presenting cells absorb antigen, activate macrophages and lymphocytes, then support the production of cytokines. An adjuvant name: incomplete Fred's adjuvants, oil and water emulsion. It's an oil and water emulsion type. The mechanism of action include delayed release of antigen, enhanced uptake by macrophage. Then complete Fred's adjuvants, oil and water emulsion with dead mycobacteria. Delayed release of antigen, enhanced uptake and introduction of co-stimulators in macrophages. Then haptens. Haptens are no molecular weight compounds that can combine with antibody but cannot initiate an immune response unless it is combined to larger carrier molecule. So haptens are low molecular weight compounds that can combine with antibody but cannot initiate an immune response unless it is coupled to a larger carrier molecule. Haptens are low molecular weight compounds that can combine with antibody but cannot initiate an immune response unless it is coupled to a larger carrier molecule. A substance that is non-immunogenic but which can react with products of a specific immune response. Free haptens have the property of antigenicity but not immunogenicity. Then epitope or antigenic determinant, that portion of an antigen that combines with the products of a specific immune response. So always remember, epitope is a portion of an antigen. Okay. Then antibody is a specific protein that is produced in response to an immunogen and which reacts with an antigen. Then immunogen, a substance that induces a specific immune response. Immunogen have the property of antigenicity and immunogenicity. Thus, all immunogens are antigens, but not all antigens can to be immunogens. Then structure and function of antibody. Immunoglobulins, IgS, are glycoprotein molecules, also called antibodies, ABS, that are produced in response to foreign substances entering the living body. Antigens or immunogens, viruses, bacteria, toxins, etc., binding to them and forming antigen antibody complexes, resulting in AG elimination and protection of the body of the host. That is, immunoglobulins are glycoprotein molecules, also called antibodies, that are produced in response to foreign substances entering the living body. Antigens or immunogens, virus, bacteria, toxins, etc., binding to them and forming antigen antibody complexes, resulting in AG elimination and protection of the body of the host. IgS are produced by the lymphocytes and are found in fraction of blood called gamma globulin. General functions: antigen binding. IgS bind to specific antigenic determinants or ADs of an antigen. They bind to at least two or in a very few cases more ADs which can closely relate, and the number of ADs and AB can bind to is referred to as its valency. Most IgS mediate several effector functions which include fixation of complement that results to lyse of cells and release of biologically active molecules, binding of various cells to facilitate specific functions by bound cells, phagocytic cells, lymphocytes, platelets, etc. Then most effective functions of ABs are carried out after the AB binds to AGs. Different IgS molecules can have different AG binding properties because of different VH and VL regions. Then structure of immunoglobulin. All IgS have the same basic structural unit of two identical light chains and two identical heavy chains. The heavy chain and light chains are joined together by interchain disulfide bonds and non-covalent interactions. The number of interchain disulfide bonds varies among different IgS. So within the polypeptide chains, the heavy and light chains, there are also present intrachain disulfide bonds. So amino acid sequence of both heavy and light chains of an Ig characterizes two distinct regions of the chains based on variability of the amino acid sequence, known as variable V and constant C regions. Light and heavy chains are composed of both a variable and constant region designated VL and CL light chains and VH and CH heavy chains. The amino acid sequence of the variable region form the N-terminal ends of the chain and determine antigenic specificity of the Igs. Constant region are the same for each specific class of Ig and carry the effector sites. Then light chain, we are going to specifically learn about light chains. VL about 100 to 110 amino acids, CL 100 to 110 amino acids. There are two types of light chains, kappa and lambda. The kappa are twice as much as lambda. There are also four classes of lambda chains. These chains were about 23 kilodalton. Differences in the type of light chain also form a basis of grouping of IGs into various types. The variable region makes up half of the entire light chain and the constant region the remaining half. Then heavy chain. VH 110 amino acids, CH 330 to 440 amino acids. There are 5 types of heavy chains which define the class of IGs, namely alpha, gamma, mu, delta, and epsilon. Alpha, gamma, mu, delta, and epsilon. Heavy chains are between 53 to 75 kilodalton. The variable region makes up a quarter of the entire heavy chain, while 3/4 of the remaining chain is the constant region. The hinge region is the area of Ig where the arms of the ABs form a Y. It is flexible region. IGs also have domains formed from flukes of the globular region containing the interchain disulfide bonds, and they are VL and Cl, light chain domains, and VH and CH, heavy chain domains. Seen in the three-dimensional images of Ig, the constant region of light chain and the appropriate heavy chain form globular constant domains, while the variable region of light chain 1 and corresponding heavy chain interact to form globular variable domain. Immunoglobulin types and classes. Based on differences in the amino acid sequence in the constant region of the heavy chain, there are 5 classes of Igs: IgG, gamma heavy chain; IgM, MIU heavy chain; IgA, alpha heavy chain; IgD, delta heavy chain; and IgE, epsilon heavy chain. IgGs are monomers. Subtypes and subclasses differ in number of disulfide bonds and lengths of hinge region. Then IgA, serum IgA is monomeric, but IgA found in secretions is a dimer having a J chain. So secretory IgA also contains a protein called secretory piece or T piece. This is made in epithelial cells and added to the IgA as it passes into secretions, helping the IgA to move across mucosa without degradation in secretions. Then IgM, it normally exists as a pentamer in serum but can also occur as a monomer. It has an extra domain in the mu chain. Then and another protein covalently bound via S is called J chain. The chain helps it to polymerize to the pentamer form. Then IgG properties we are going to learn. It is most versatile Ig and can carry out all functions of Ig molecule. It is major Ig in serum. It is also found in major Ig in extravascular spaces. It is only Ig that crosses the placenta. It flies complement, although not all subclasses do this well. It binds to cells and is a good poisoning substance that enhances phagocytosis. Then IgA properties. It is the second most abundant Ig in serum. It is the first class of Ig in secretions, tears, saliva, colostrum, mucus, and is important in mucosal immunity. When it binds to some cells, PMN cells and lymphocytes, it does not normally fix a complement. The next IgM properties. It is the first Ig to be made by fetus in most species and new B cells when stimulated by Ags. It is the third most abundant Ig in serum. It is also able to bind some cells via Fc receptors. B cells are surface IgMs which exist as monomers and lacks J chain but have an extra 20 amino acid at the terminal that anchors it to the cell membrane. Then IgD, it exists as monomers. Properties, it is found in low levels in serum and its role in serum is uncertain. It is found primarily on B cell surface and serves as a receptor for Ag. It does not fix a complement. Then IgE, it occurs as a monomer and has an extra domain in the constant region. It is least common serum Ig, but it binds very tightly to Fc receptors on basophils and mast cells even before interacting with Ags. It is involved in allergic reactions because it binds to basophils and mast cells. It plays a role in parasitic helminthic diseases. Serum levels rise in these diseases. Eosinophils have Fc receptors for IgEs, and then when eosinophils bind to IgE coated helminths, death of the parasite results. So antigen binding site is there, and we try to discuss the typical structure of an immunoglobulin. We could see antigen binding sites will be there, light chains will be there, variable heavy chain and variable light chain regions. Hinge region will be there, Fab region, heavy chains, carbohydrates are there, like that.